Your immune system is like the military,
Hey, yo.
with two main branches, the innate immune response and the adaptive immune response. The innate immune response is immediate and nonspecific, meaning that although it can distinguish an invader from a human cell, it doesn't distinguish one invader from another invader. In contrast, the adaptive immune response is highly specific for each invader. And that's because the cells of the adaptive immune response have receptors that differentiate friendly bacteria and potentially deadly ones from their unique parts, called antigens. This adaptive immune response takes days to weeks to become activated, but is also responsible for immunologic memory. Now, the key cells of the adaptive immune response are the lymphocytes, the B and T cells, which are generated during lymphopoiesis. Lymphopoiesis has three goals. First, to generate a diverse set of lymphocytes, each with a unique antigen receptor. Second, to get rid of lymphocytes that have receptors that are self-reactive, meaning that they'll bind to healthy tissue. And third, to allow lymphocytes that aren't self-reactive to continue maturing into secondary lymphoid tissue. Normally, hematopoietic stem cells within the bone marrow mature into a common lymphoid progenitor cell, which then becomes either a B cell or a T cell. To become a B cell, it has to develop into an immature B cell in the bone marrow, and then complete its maturation into an antibody-secreting B cell, called a plasma cell, in the lymph nodes and spleen. To become a T cell, it has to migrate to the thymus and become a thymocyte, where it completes its development into a mature T cell. So B for bone marrow and T for thymus. In T cell development, the common lymphoid progenitor leaves the bone marrow and goes to the thymus, because that's where the developing T cells or thymocytes mature. The thymus is a fatty organ that sits in front of the heart, and as we age, much like our waistlines, the thymus gets fattier and fattier. This fat crowds out the space that used to be reserved for T cell development, and it's one reason why cell-mediated immunity declines over time, which is a process called involution of the thymus. So the thymus has an outer cortex and an inner medulla. Within the medulla, there are epithelial cells which support the developing T cells, as well as dendritic cells which present antigens to developing T cells on molecules called major histocompatibility complexes, or MHC molecules. That's important because T cells have T cell receptors that can only bind peptide antigens if they're displayed on an MHC molecule, which is sort of like a silver platter. So if the T cell receptor can't bind to the MHC molecule, then the T cell isn't going to be able to do its job. So of course, a key element of T cell development is formation of this T cell receptor, which is made of two chains. An alpha chain, which is like the light chain of a B cell receptor, and a beta chain, which is like the heavy chain of a B cell receptor. The alpha and beta chains are made up of gene segments called V for variable, D for diversity, and J for joining. The beta chain includes one V segment, one D segment, and one J segment, while the alpha chain only contains one V segment and one J segment. Every person inherits multiple copies of the V, D, and J segments, and they can be rearranged interchangeably to make a unique structure. This is kind of like how you might have several pairs of shoes, pants, and shirts, and can mix and match them to create lots of different outfits. For the alpha chain, each person has between 70 and 80 variable or V gene segments, and 61 joining or J segments, and that's just for the alpha chain. There are more V, D, and J segments for the beta chain. So one T cell might have a beta chain on its T cell receptor that has a V1, D3, J5 combination, and an alpha chain that's V7, J2. And another T cell might have a beta chain that has a V44, D10, J1 combination, and an alpha chain that's V2, J3, thus making completely different T cell receptors with completely different antigen specificities. Now, to have a fully functioning T cell receptor, a T cell has to get through a series of successful gene rearrangements, first for the beta chain and then the alpha chain. And if the T cell fails at any stage, it dies.
Blech.
T cell development is broken into 3 key stages, based on which key molecule it's expressing on its surface at any given time. These molecules, CD3, CD4, and CD8, are expressed in addition to the T cell receptor. All T cells express the CD3 molecule, which is part of the T cell receptor. And once the T cell is fully mature, it'll either express CD4 or CD8. When the common lymphoid progenitor first arrives in the thymus, it doesn't express anything on its surface, so it's considered CD3 negative, CD4 negative, and CD8 negative. And it's referred to as a double negative stage or DN stage cell, because it has neither CD4 nor CD8 at this point. The DN stage can be further broken down into DN1, DN2, DN3, and DN4. As the cell moves through the steps to create a functional T cell receptor, it'll then begin to express CD3 in addition to CD4 and CD8, all on the same cell. The dual expression of CD4 and CD8 is why it's called the double positive or DP stage. Once the cell completes its expression of a functional T cell receptor, it'll continue to express CD3, but then it'll downregulate expression of either CD4 or CD8, and be known as a single positive T cell, or SP. Throughout all of these stages, the T cell interacts with epithelial cells of the thymus, which release growth factors as well as molecules like interleukin 7 and interleukin 2, called IL-7 and IL-2 for short. And these make the DN1 cells mature. 2 enzymes, RAG1 and RAG2, start getting expressed, and that signifies that the cell is now a DN2 cell. To start forming the beta chain, RAG1 and RAG2 help to splice together D and J segments on both chromosomes. And the chromosome that successfully rearranges first will then suppress the other chromosomes from rearranging, a process called allelic exclusion. If a cell successfully joins a D segment to a J segment, then it's considered a DN3 cell. Next, the DN3 cell has to attach its DJ gene segment to a V gene segment, with the help of an additional enzyme called VDJ recombinase. Once the VDJ segment is combined, the full beta chain's antigen binding site is complete, and needs to be recombined with the constant region of the T cell receptor. Once that's complete, it's considered a DN4 cell. At this point, it's time to test out if the beta chain is functional, by seeing if it can bind to an invariant pre-T alpha chain, and get expressed on the cell surface. The invariant pre-T alpha chain is basically something for the beta cell to practice with, until the real alpha chain is eventually made. So the beta chain, the invariant pre-T alpha chain, and CD3 are all expressed on the surface together, and they're joined by another beta chain, invariant pre-T alpha chain, and CD3. The two complexes join together to form a homodimer, and that sends a signal down the cytoplasmic tails of the receptor to the cell's nucleus. When the signal is sensed within the cell starts to divide, and the daughter cells of the rapidly proliferating DN4 cells are called DP cells, because they start to express both CD4 and CD8 on their surface. The DP cells now start rearranging the alpha chain, which means that all the DP cells will have the same beta chain, but each will have its own distinct alpha chain. As long as it creates an alpha chain that can bind to the beta chain and is not self-reactive, the DP T cell moves on to the next step. If it doesn't, the developing T cell dies. Blech. Now, T cells undergo a process called central tolerance, which is a quality control process that makes sure the T cell can recognize and bind to MHC molecules, a step called positive selection, but that they don't bind too strongly to become self-reactive and attack self-tissue and cause disease, a step called negative selection. The T cell receptor has a couple different parts. One part forms the antigen binding groove. And surrounding the antigen binding groove is the invariant region, which has to bind to the MHC molecule. In fact, MHC molecules also have an antigen binding groove and an invariant region. So the invariant regions on both the T cell receptors and the MHC molecules bind to each other. In positive selection, only the DP cells that bind to an MHC molecule are allowed to survive. Those that don't are killed off. In negative selection, to help identify self-reactive T cells, there's an autoimmune regulator gene, called AIR for short. AIR is expressed in primary lymphoid organs, and allows them to express antigens which are normally found all over the body, like the brain, ovaries, pancreas, and so on. It allows these primary lymphoid organs to serve as a microcosm of the entire body, kind of like how the Epcot Center theme park in Florida is a microcosm of the world. So that allows the body to test out these newly formed T cell receptors in a safe environment. If a DP cell binds strongly to a self-antigen, then it dies swiftly and silently by apoptosis. Whereas if a DP cell recognizes self-MHC but doesn't recognize the self-antigen presented in the MHC molecule, it'll go on to become a single positive naive T cell by downregulating either the CD4 or CD8 receptor. The question is, which should it downregulate, CD4 or CD8? This is determined by how strongly the T cell receptor binds to the invariant regions of the MHC molecules. Whenever a T cell receptor and MHC molecule bind, CD4 binds to the MHC class II molecule a bit like an arm that reaches out and holds the T cell receptor and MHC together. So if there's a DP cell, its CD4 will bind to an MHC class II molecule. Now, if the strength of the binding between the T cell receptor and the MHC class II molecule is somewhat strong, then the cell will downregulate CD8 and become a single positive CD4 positive T cell. On the other hand, if the strength of the binding between the T cell receptor and the MHC class II is somewhat weak, then the cell will downregulate CD4 and become a single positive CD8 positive T cell. At this point, the cell is considered a single positive naive T cell that'll travel to the secondary lymphoid organs like the lymph nodes or the spleen to deal with antigens presented on MHC molecules by antigen-presenting cells like dendritic cells. Alright, as a quick recap, T cell development happens in the thymus and has a few key tasks. Rearrange the beta chain, rearrange the alpha chain, make sure that the T cells bind the MHC molecule, called positive selection, and make sure that the T cells aren't self-reactive, called negative selection. The beta chains first combine DJ segments, then the VDJ segment forms, and finally a constant region is added on. A homodimer forms with two copies of the beta chain, the invariant pre-T alpha chain and CD3 molecules. At that point, the cells proliferate and each daughter cell rearranges its alpha chain, combining a V segment to a J segment. The alpha chain will then join the beta chain and undergo positive selection and negative selection to make sure that they can bind MHC molecules but are not self-reactive. At this point, it becomes either a naive CD4 positive T cell or a naive CD8 positive T cell, and is released into the periphery.